Hello, and welcome to the VectorCat platform. VectorCat allows researchers, data specialists, and business users easily find data in their organization and draw insights from it. For instance, let's search for a sequencing protocol. The platform searches through the names and contents of the document semantically. Here it found something named protocol NGS and gave some highlights. Let's say I want a brief summary of the protocol to better understand if it's relevant for me. AI Assistant summarizes the protocol. Now I can see the main steps and get the sense of the relevancy of this result. That can be very useful, especially if the document is very long. Let's go and find other sequencing files. In a real company setting, there will be hundreds of thousands of those, so using filters on the right might be helpful. If I'm interested in the final data that I know will be in dashboards, I'll filter by sources and find some sequencing results in Power BI. So let's open this data. A typical problem in a company would be that even though I may have access to the final data, I don't necessarily know how it was acquired and why these quality metrics look the way they do. Instead of writing to someone in Teams, I can use our data maps module to avoid this. By using a browser plugin, I can go to the data map associated with this chart and realize that it's just a final step in a pretty long sequence of experiments that originated back in the lab. Then they were processed in AWS S3 and Microsoft Fabric, and finally ended up here. Each box on this map relates to a separate experimental step, or data processing, and provides the main information about the asset, such as its owner, date, and so on. If my permission allows, I can go directly to each of the assets and explore the details. Like, for example, here in BenchLink, I can see the designs of the plasmids and the mutations that were introduced. But sometimes it's just not efficient to manually explore all the processing steps. For example, for complex omics experiments, researchers spend hours manually annotating the data, as they need to extract dozens of parameters for dozens of samples to run analysis pipelines correctly. And we've automated this process, reducing hours of manual work. Let's say I want to add metadata to the dashboard we have just seen, and I can choose from a template that I previously created on a platform. In this case, I've chosen the next generation sequencing, and I will ask the agent to fill in this form for me. The agent is doing its job, displaying what's going on the backend. What actually happens is it goes through all the assets connected to the dashboard, which I have shown you on data maps, explores them, and extracts the relevant values. It fills in the values that it can find or infer via logical steps, and provides it to the user for validation. Importantly, if it has to make any assumptions or conversions, it documents them to make the process transparent. For example, here, it converted the units. And even more importantly, it doesn't fill in the values that were missing from the documentation and highlights them in red instead, drawing the user's attention. Let's say I'm happy with the outcome. I can save this form, and it will become attached to the asset. This data can be further accessed and consumed via our API, or I can use it to find the assets I'm interested in our platform more precisely. These forms for custom metadata can be added or edited in the platform on the go, in the builder, which looks a little bit like Google Forms. And it is integrated with the search module to provide additional filters. Now that I'll search for sequencing files on our platform, there is a custom filter block that will appear. The values relevant for the query will be automatically populated. For instance, I can filter for samples with hex cells, and it will return me the dashboard we were previously looking at, because now it has this metadata.